for that introduction, and thanks for having me here. It's awesome to get invited to Dauphin Island. I got to pack up my wife and my baby and her in-laws, and we're all here now for the weekend, so we get to make a weekend out of it. But thanks for the invitation, and thanks for coming to my talk today. I'm going to be talking about some of the work that my lab is doing, really focusing on the persistence of marine metapopulations in a changing ocean. Okay, just some background on me, just to add what was just said. I'm originally from upstate New York. I'm from Syracuse. And I did my undergraduate and graduate work at the University of Miami, where I studied mostly deeper coral populations, what are sometimes called mesophotic reefs, and their reproduction and connectivity to shallow environments, and the potential that those deeper reefs could serve as refuges during climate changes. I continued that work when I was in at the University of the Virgin Islands, and I started to think about models of metapopulations to understand how persistence and extinction might happen in the next century. And then I was at the Duke Marine Lab for a while and eventually landed at Louisiana State University, where I have a stellar group of students who work with me, and I want to acknowledge them now, but throughout the talk, because they've contributed significantly to everything I'm going to talk about today. I'd say that I was trained as a coral reef ecologist, and I still consider myself a coral reef ecologist, although we focus in the lab on other types of marine populations. And I became a marine ecologist or a coral reef ecologist because I was really fascinated by coral reefs. They're sort of endlessly dynamic and complex and beautiful. But if you're a coral reef ecologist today, you really are forced to grapple with the reality that many organisms on coral reefs may be facing extinction in the next century due to climate changes and other stressors. And this is a real bummer for someone who loves coral reefs. And so most of my research revolves around this idea of persistence and what makes a population or a metapopulation persistent in time and space. A lot of that research focuses on the dispersal of propagules in between distinct populations. This includes, for the most part, larvae, marine larvae, and how they get from place to place. But more and more, my lab has also been looking at the dispersal of pathogens in between patches or populations, and how that can influence the persistence of metapopulations. So this is, there you go. This is sort of the lab's current, I'm not going to say model, of sort of a Venn diagram of how we sort of try to get at persistence. So we study the reproduction of invertebrates, mostly corals, and we look at reproduction in a variety of environments. We also study the connectivity and migration of larvae, but also of pathogens, and also perturbation to population persistence like climate changes, coastal development, and also recently disease. And then also potential refuges from those disturbances and how that all influences persistence. My students are sort of tackling all angles of this Venn diagram, and we have funding to sort of look at persistence piecemeal. Hold on a second. I'm going to be talking about 3 distinct but interrelated studies today. The first one is at sort of the regional scale of the Western Atlantic or Caribbean, and looking at the persistence of a coral's metapopulation in a changing ocean over the next century. And then the next study, I'll zoom in a bit closer on an individual coral reef patch and how larval behaviors and coastal flow can influence the retention of larvae near their natal habitat and how that can influence local persistence. And then the last study I'll be talking about is an emergent disease in the Western Atlantic called stony coral tissue loss disease, which some of you have probably heard of. It's a really nasty new disease that's been causing an outbreak for about 8 years. And I'll be talking about how the lab is doing modeling at various scales to understand how this disease is moving through populations. So this is just an outline of today's talk. I'm going to be talking a bit about just some background on dispersal of propagules in the ocean and metapopulations, and then I'll talk about those 3 studies. And it's kind of the opposite of a bad news sandwich. I'm giving you kind of a good news sandwich. And that's just kind of what you get when you study coral reefs these days. Most of it is bad news. So I tried to sandwich some good news. So I've said the word metapopulation a couple of times, and I want to make sure everybody understands what I'm talking about. I've sort of nested this inner circle that represents a local population here. And at the scale of the local population, the persistence is based on the number of new individuals coming into the population, usually through births, but if you're a coral, also through asexual propagation. And then deaths. And so if you have more births than deaths, then that population should have population growth and persistent time. This is the scale that most ecological studies are sort of necessarily done at. But we acknowledge that new individuals can come into the population through immigration and leave through emigration as well. And so this local population is nested through connectivity in a metapopulation. So each of these circles could represent a coral population or patch, maybe a reef, and has internal processes going on with births and deaths, et cetera, but then can also be connected to other patches through larval dispersal. So individuals produced here can leave and disperse in the ocean to other reefs, or a coral reef can receive individuals from an upstream reef. What that means is that these population processes aren't really decoupled from other populations in the network. And what I mean by that is that if this reef experiences some sort of perturbation or disturbance, like a hurricane or a disease, it affects the number of emigrants that they're putting into the larval pool that can then subsidize recruitment downstream. So you can imagine that perturbations can actually propagate through this network. So just to sum this up a little bit, most marine and coastal populations, especially benthic and demersal populations, are fragmented in this way. And most organisms that live in these sorts of habitats also disperse as larvae. And so it's networks of these connected patches form metapopulations. And it's at the scale of the metapopulation that persistence or extinction really occurs. And I hope that you will leave today believing me that a population's response to environmental change both influences and is influenced by how it's connected to other patches in the network. Most larvae, and certainly pathogens, are small and they usually lack hard parts, and they're actually very difficult to track in the natural environment in situ. And for that reason, my lab employs a suite of modeling tools to try and understand how patches are connected to each other. And for the most part, we're using Lagrangian particle tracking models. Now, these models, really what they just involve, they involve a model of the ocean, of ocean currents, but also other parameters of the physical environment. And we drop virtual particles that represent, for example, larvae into that ocean and see where they go. We can give those larvae all sorts of traits and behaviors that try and make them more and more realistic. And my lab is also really committed to doing both lab and field work to make sure that we're giving these larvae realistic behaviors, for example, and that we are realistically representing the populations that we study in these models. So I'm going to talk about the first study of the talk. This paper came out just a couple of weeks ago, and it really asks the question, how does environmental change influence migration and persistence, in this case, in a Western Atlantic coral? I'm sure everybody in this room is familiar with coral bleaching, but I will tell you about it just in case. Coral bleaching is a breakdown of the symbiotic relationship between a coral host and its endosymbiont dinoflagellate algae. So this algae photosynthesizes and produces sugars that the coral uses for energy. And when it gets too hot, for example, that the photosynthetic process in the algae stops working correctly, and essentially the coral can boot the endosymbiont out of its tissues. And the tissue will, well, it'll look like the coral turns white, but what happens is the algae gives the coral most of its color. And so we're seeing right through the jelly-like tissue of the coral to the white skeleton underneath. Now, if temperatures return to normal or cool off soon enough, the coral can usually recover from this. But if it doesn't, or if the coral experiences sequential bleaching, the coral can starve and die. Now, we know that coral bleaching is increasing in severity everywhere, essentially, we look. But we also know that different locations and different coral species have different risk of coral bleaching. So we're actually pretty good at predicting whether or not corals will bleach in any given place, for example, in a year. But what we don't really understand is how this heterogeneous risk of coral bleaching will influence the metapopulation as a whole. And so we set out to at least try to chip away at this question by merging together a couple of different models. The first one is a thermal projection model. So this model predicts what the temperature in the ocean will look like over the next century or so. And it also goes a step further, and it predicts whether or not those conditions are expected to be associated with coral bleaching. And then we also have a model of larval dispersal for a coral called Orbicella annularis, which is a threatened reef-building coral in the Western Atlantic. And we merge these together into a metapopulation model, which for each decade in the model essentially asks the question, is the metapopulation persistent given what's happening at each patch? So this is the distribution of thermal stress in that thermal projection model for each decade of the model. On the x-axis here, we've got essentially the thermal stress that each patch experiences. If it's a 1, that means that the model predicts that corals are going to bleach in that location every year in a decade, which is really bad news for a coral. And we can pretty much say that that would be potentially local extinction for corals if they're experiencing this. Obviously, further on the left is lower stress, and each of these lines is a decade. But I want to focus on the middle of the century, where the distribution goes something sort of like this, where there definitely are some reefs experiencing extreme thermal stress, but most of them are towards the left-hand side of this graph. And within about 10 years, the distribution goes to something like this, where most coral reefs are experiencing extreme thermal stress, but there is this long tail to this distribution, suggesting that some reefs are thermal refuges. And so again, the metapopulation model is just asking, okay, do we expect each patch to be, for example, reproducing and putting larvae into the ocean where they could then disperse to other patches, or do we expect that coral reef to die and experience recruitment failure and not provide larvae downstream? Unfortunately, what you kind of expect to happen happens in the model anyway, where around the middle of the century, the metapopulation becomes unstable. So here on the x-axis, we have the decades in the model. The y-axis is something called metapopulation capacity, which is a measure of whether or not a metapopulation is persistent. And what you need to understand is just that if a metapopulation dips below this threshold of 1, it means that the metapopulation has entered into an unstable equilibria and is potentially going to go extinct. That metapopulation is shrinking and will eventually cease to be persistent. We also, you'll see a bunch of lines here, and those different lines represent, if we, in the model, say that individual patches are more or less resistant to that thermal stress. Because of some of this, we don't really know. So we did a sensitivity analysis. And essentially, even if coral reefs are individually very resistant to thermal stress, we still predict that the perturbation to connectivity caused by this thermal stress still drives essentially metapopulations that are very resistant into unstable equilibria. So that's crappy news. And I stress that this is a model based on a whole bunch of assumptions, but the model does suggest that the way that we understand thermal stress to be distributed in the next century or so is bad news for this coral. But this is the exact same. So saying that, there's a lot of information we can extract from this model that might be useful to mitigate that future. So this is the same result, but shown spatially, graphically, rather than the figure you just saw. So each panel is a decade in the model, going through time here in 2 columns. And I'm going to just talk about the different components of this figure, and then we'll zoom in, because I know it's a little difficult to see. What you can see is that these individual patches have a different color, and that color represents their thermal stress in that decade. So cyan means cooler reefs, and magenta means hotter, stressed reefs. And you can also see that the size of the circle that represents these patch changes, and bigger circles are what's called more central to the connectivity network. And what that means is that they're facilitating connections to other reefs. And so they are becoming more and more important for metapopulation functioning. And you can see some gray lines. Those represent larval dispersal. And you can also see these hulls that we've drawn around reef patches. And that's the spatial extent of the metapopulation. And so you can see that over time, the metapopulation fragments into smaller metapopulations. You can also see the color of the hull changes. And as soon as it goes into the red, that means that it's fallen below that threshold where those metapopulations are unstable. So I'm going to zoom in here on the middle of the century, right before things appear to get really bad. And you can see a lot of magenta reefs. Those are stressed reefs. And you can see also some cyan reefs, which are those thermal refuges that I was talking about. You can also see that the size of the patches, or the centrality of the patches that are cooler, are bigger. And so what that means is that they are more central and are sort of carrying the load for metapopulation persistence. If we follow those reefs through the next couple of decades, what we see is that the metapopulations collapse around those thermal refuges. And essentially, smaller metapopulations are centered around those refuges that are facilitating larval exchanges because they're still persistent. So there's some implications here for management and restoration. I think that using models like this one, we can start to understand better where to allocate, for example, restoration or management activities. In particular, outplanting activities, which are very expensive, putting them in places where we expect connectivity to still be occurring, or dispersal to still be functional, is important. If you can mitigate temperature stress, I don't know if you can on large scales, but if you can, these also might be areas to focus on. But certainly it would make sense to mitigate other stressors, because this is just thermal stress that we're looking at. We know that coral reefs suffer from eutrophication and overfishing and other stressors. So if we can reduce those synergistic stressors at these reefs, it might be important. It's also potentially a place for us to look for potentially resilient or resistant corals or symbionts, because these are areas that are going to be experiencing temperature stress for a longer duration and potentially persistent for a longer duration. I also think that this model may show us the environmental conditions beyond which coral reefs in the Atlantic may not be able to persist. And so I think that's an important thing for policymakers to understand when we're trying to figure out how to reduce emissions, or if we're going to be able to reduce emissions. This may be representative of conditions that we should be aiming not to surpass. So I'm going to switch gears slightly and hopefully tell you some better news. We're going to zoom in on an individual coral reef patch or patches and try and understand what it is about the physical environment and also the behavior of coral larvae that can limit their advection away from natal habitats and allow them to contribute to local persistence. So I'm going to introduce you to the island of Moorea in French Polynesia. Has anybody been to Moorea? I know at least you have. This is the site of a NSF long-term ecological research program. So there's a lot, coral reef researchers have been studying coral reefs here for a long time, and hopefully will be for a long time as well. And although these reefs are isolated and beautiful, they are not immune to stress. In fact, they experience periodic outbreaks of the crown of thorns starfish, which is a coral predator, and it can bloom in big population booms and eat essentially entire coral reefs. And in 2010, this happened on the North Shore of Moorea, and it was swiftly followed by a Category 4 typhoon, Typhoon Ali. And what this did was it essentially wiped the coral cover, not just the coral itself, but actually the coral structure off the reef. And coral cover went from over 40%, which, as someone who studies mostly Caribbean corals, is incredible. Caribbean coral reefs are usually like 2 to 5% cover. Anyway, it essentially wiped the coral cover of this reef down to about 0 in a very short amount of time, which is amazing. But even more amazing, I think, is that the coral cover recovered in just a few years, almost to pre-disturbance levels. That is unheard of, especially in the Caribbean. So something is happening on this reef that we should really understand, because something has made this reef very resilient. A bunch of studies have shown that a lot of that recovery was driven by the sexual recruitment of new corals. So coral larvae coming in and settling on the reef. We've done some population modeling that seems to show that the initial recovery was likely driven by the immigration of larvae into this reef from nearby reefs, which in and of itself is really cool, because that shows that this reef is nestled within a functioning metapopulation. It experienced what's called the rescue effect and was recolonized by upstream reefs. But what also is very cool is that our population modeling seems to suggest that there was a handshake that occurred during this recovery, that where recruitment went from being driven by immigration to being driven by self-recruitment or self-retention of locally produced larvae. So what we think is happening is there's a bad times versus good times relationship on this reef between immigration and retention of larvae. And that may be something that makes this reef especially resilient, because it's able to rely on immigration when times are bad, but when times are good, it's able to repopulate itself. This relates to a discussion in marine biology or biological oceanography that's been going on for a while. 40 or 50 years ago, large vessels would go out and tow the plankton and find lots of coastal larvae in the open ocean. And so the theory was that the coastal populations are probably open, and that the ocean is this bathtub full of larvae and sloshing around, and long distance connections are probable. But in the last 20 or 30 years, we've discovered that this is probably not true. Long distance dispersal, although possible, is likely not the norm. And most local populations are likely most local populations likely rely on local retention of larvae in order to remain persistent. And we know very little about how, in particular, coral reef larvae would avoid excessive advection away from their local reef. We go out and we can find coral reef larvae out in the open ocean. So they obviously do get infected, but some of them must stay close to home. And so we don't really know how. We started asking these questions when I was at the Duke Marine Lab working with Jim Hench, who's a coastal oceanographer, and his student, Walter Torres, who's now Dr. Torres. And we started looking at it from the physical side of this question. And we asked, what are the physical processes of a typical coral reef that would allow a, let's say, entirely passive particle to remain near that reef? And so here's a bird's eye view of a typical reef. This is in Moorea. And we've got the reef crest here where waves are breaking. There's a fore reef out here, and then this is open ocean. There's a back reef. There might be a lagoon back here. And this is the reef pass, where water that's driven onto the reef can then leave the reef. And so I'm just going to show a doodle of the same thing. And what we hypothesize is that there's got to be sort of a relationship between the alongshore current that's driven by, the onshore current that is driven by waves, the offshore current through this reef pass, sometimes we would call this the reef jet, and the alongshore current. And if these are in the right balance, some particles might be able to recirculate over the reef in a fore reef, back reef circulation cell. Some others, in slightly different conditions, would be affected along the fore reef or maybe out into the open ocean. So we started testing out these ideas in a toy model. So an idealized coral reef that has those same features. It's got a reef crest, a fore reef, a back reef, a lagoon, and reef passes. And we varied the alongshore current, the wave height, and the bottom roughness to look at the sort of Goldilocks conditions that would result in these fore reef, back reef circulation cells. And so we're still working on this, but finding that those Goldilocks conditions are within the ranges of wave height, alongshore current, and bottom roughness that you'd actually find on a coral reef. One thing I will mention just briefly, another thing that's really cool is what this suggests is that very energetic reefs with lots of waves and alongshore current actually might be more retentive to larvae than we initially would have thought. I would have thought that a high alongshore current, for example, would eject particles away, and they do, it does, but if that alongshore current is interacting with the reef jet, it bends those particles back towards the reef and allows them to sort of surf the Stokes drift back into the back reef. And I can talk more about that later, if you'd like. So we're applying these same ideas in Moorea. This is the north shore of Moorea, and we have the beginnings of a working model. And we're doing the same thing, releasing passive particles, and we find a lot of those same structures, and actually some additional structures that would retain passive particles for the same amount of time that a coral larvae would take to develop and potentially need to settle out. The cool thing about doing this in Moorea is there are all these long-term data sets about the coral community, about the corals that are spawning, and about recruitment that we can use to parameterize and also to validate these models moving forward. One hole in the data sets that we are looking to fill is that coral researchers have been looking at coral larvae for a long time, collecting coral larvae, putting them in a tank, and recording what they do. The resolution in terms of how often they're taking measurements, it's usually like every 12 hours or every day, and the sorts of behaviors they record is like, okay, the particles are up at the top of the tank, or at the bottom of the tank, or they've settled, or they've died. And we're discovering that this isn't really the kind of data that we need to be able to parameterize these models. These physical models have a time step of less than a second. So we really need to have an understanding of what these larvae are doing on sort of a similar time scale. And so my student Ben Limer and I are trying to do that. We're 3D printing larval dispersal chambers that rotate, and they're connected to a servo motor and an Arduino controller, and they rotate every minute or so to keep those larvae from settling out at the bottom or the top of those columns. And then after they rotate, a video turns on and records the larval traits or swimming. And then we feed that video into a computer vision algorithm that estimates their swimming velocities and records their behavior. And we're still working on this. In fact, my student is in Fort Lauderdale right now doing this with a coral called Parietes asteroides. What I'm showing here is some early results for two species of coral. This is Orbicella fabulata and Montastria cavernosa. These corals actually reproduce on the same evening. They live on the same reefs. They have the same or similar depth distributions. They have a slightly different mode of breeding. One is a hermaphrodite, the other is gonochoric, so it has different sexes. So they're a little bit different. When we track their larvae over time, so this is a time on the x-axis, and this is swimming velocity or floating or sinking on the y. And so above this dotted line, those larvae are moving upwards. Below the line, they're swimming or sinking downwards. And you might look at these distributions and think, oh, well, they overlap a lot, so they're probably pretty similar. But when we do a really simple, the goal is to be able to go through these models and figure out what is the distribution of their swimming or sinking over time. And when we do a really simple model with these two species and allow a virtual particle to select from these distributions through time, what we actually see is that in a hypothetical water column, like an infinitely long water column, that we would expect total sorting of these two species vertically. So one species would be higher in the water column than the other. And this is actually pretty important for dispersal because most coastal currents are vertically stratified. And so these larvae are essentially depositing themselves into different currents of different speeds and potentially in different directions, which can affect their dispersal very early in their life. We're beginning to try and parameterize our dispersal models with this information as well, but we're just getting started. Here's some examples from, this is a Pacific species, this is a Pacific species, this is actually not, this is an Atlantic species, but we have some data on it. These left two species are brooders, so that means that their larvae are competent much earlier and can have, for example, downward swimming behaviors. And then this species is a broadcast spawner, and so it sort of necessitates that the fertilized zygotes float at the surface for a period of time. And we do see that it makes a difference on the scale of the reef. And we expect in the first 5 to 10 days of dispersal to find these larvae in different places. But we do see some retention even of a broadcast spawner on the scale of the reef. Just these top panels are basically density distributions. So hot red colors mean that we would expect to find more larvae in that place. It also determines where on the reef adult colonies should be situated in order for those larvae to be retained locally. So we see a very different distribution for the brooders as opposed to the spawners, which need to be situated in specific places on the reef in order for those larvae to be retained for a period of time that relates to coral settlement. That has implications for all sorts of things, but also for restoration. So if you're going to be outplanting a broadcast spawning species and you want some of those larvae to be retained locally, you actually have to be thinking where on the reef you're going to put them. Okay, just to sum this up a little bit, we're just beginning to understand the biophysics of retention of coral larvae near reefs. It appears that larval behaviors are very important, and this really builds on some work that was done with oyster larvae and crab larvae that looks at selective tidal stream transport. It's a little bit different just because the environment is very different. These aren't really as tidally influenced spaces, and so these larvae are not changing their behavior based on tide or the moon. They're changing their behavior just through their development. We're looking at this experimentally in Florida and also hopefully Moorea soon. We also just briefly, we were funded by NSF last year to do a very similar set of studies with stone crabs and their larvae. My colleague discovered that stone crab larvae reverse their vertical swimming under low pH conditions. And so We're putting stone crab larvae into an array of future ocean conditions, low pH and high temperature, to understand how it influences their survivorship, but also their behaviors. And we're building dispersal models to look at how it affects this crab's populations in Florida. I'm happy to talk more about that later too, if you'd like. I'm going to switch gears a little bit again and talk about my third study, looking at an emergent disease, coral disease in the Western Atlantic. And we're really applying a lot of the same tools, dispersal modeling in particular, to understand how connectivity influences the trajectory of this disease and how this outbreak has played out. Swimming coral tissue loss disease showed up on the Florida Keys in 2014, so it's been around for about 8 years now. It quickly moved through the Florida Keys, has caused a lot of damage. It then later showed up throughout the Caribbean and still showing up in new places. It's the worst coral disease in recorded history, and different species, actually over 22 coral species are affected by this disease, but these different species have differential susceptibilities to the disease. They can have the disease for a different amount of time, and they experience different rates of mortality. Here we're scuba diving on a reef in the US Virgin Islands. This is from 2019. You can see these large lesions associated with the disease. Some of these corals are completely dead. This disease can kill corals very quickly. And it's a bummer. It's a beautiful, beautiful reef. So from early on, it appeared that this disease was dispersing on ocean currents, whatever was causing it. It showed up here near Miami, and then spread down the Florida Keys and eventually to the dry Tortugas. So we thought, okay, we can use a lot of our same tools to address the dispersal of this disease. Oh, I want to add that even though it appears to move on ocean currents, it's also popped up in areas in the Caribbean that it doesn't really make sense hydrodynamically that it dispersed there on ocean currents. And so we think it's waterborne and that it's potentially being transported through ballast water of ships. There are challenges to studying coral disease. The first is that coral-associated microbes are notoriously difficult to culture in the laboratory. And because of this, the etiological agents of many coral disease are either unknown completely or hotly disputed. And part of that is that you can isolate microbes that are associated with disease in one place, and then you can sample a coral in another place and find a different set of microbes associated with that disease. Oh, and I will add that we do not know what is causing stony coral tissue loss disease. There are a lot of theories. It appears that antibacterial ointment can stop the spread of a lesion, but there's also seems to be some evidence that it could be a viral infection of the symbionts. We really just don't know yet. Another challenge to studying coral disease is that disease is likely transported through water or through sediments, not through the movement of individuals. And I say this is a challenge because most epidemiological modeling involves or assumes interactions of individuals. So there are disease reservoirs like the ocean and sediments that we need to account for. It's also difficult to know what exactly a coral individual is. This is a coral that has, oh, I should mention, I've been calling it skittled. Some people call it stony coral tissue loss disease, SCTLD. I just say skittled. Anyway, it's infected with this disease. You can see the lesions, but corals are colonial animals. Certainly this colony is affected by the disease, but not every individual polyp is affected by disease. And again, most epidemiological modeling is done on the scale of the individual. And so it's a little difficult to say if every individual in this picture is affected by disease. Okay, I should note that my lab is a part of some big collaborations looking at this disease. We've gotten an NSF Rapid, we've gotten an NSF Ecology and Evolution of Infectious Disease grant, working through the Department of Environmental Protection in Florida. We've got some more support, and also from Louisiana Board of Regents. So I want to acknowledge that got some big groups of excellent people working on this, and already some papers coming out about the disease. We've looked at the community responses, both the coral and microbial community responses to this disease. We've done some dispersal modeling of the dispersal of the disease, and we've also done some transmission experiments looking at how community influences transmission. My lab's role in all of these collaborations is mostly to apply, again, these dispersal modeling and epidemiological models. But also I should mention that we're doing quite a lot of histopathology. I have a student, Ashley Rosen, who's doing a ton of histopathology to try and understand how this tissue is affecting how this disease is affecting coral tissues and how the disease is manifesting in different species. One way that we try to understand how diseases move through populations is through what are called SIR models. And at this point in the global pandemic, I imagine everyone has seen some sort of SIR model. These are compartment models. SIR stands for susceptible, infected, and recovered. There are a lot of variations on these models. And essentially, they're just a bunch of differential equations that explain the rates of moving in between these compartments. So, you know, at what rate do susceptible individuals become infected, and at what rate do infected individuals recover or die from this disease? I crossed out immunity here, but put a question mark because we don't currently have evidence that corals can recover from this disease with immunity. Corals have very rudimentary immune systems. But we are finding that it's getting harder and harder to infect corals in our transmission experiments. So it appears that either the corals or the microbial communities or the endosymbiont communities or some combination are not naive to this disease, at least in the US Virgin Islands. So that's why I crossed it out, put a question mark. Anyway, from the scale of the metapopulation though, we know that there's probably internal dynamics where this disease can move in between corals on a coral reef. And there's also metapopulation dynamics where the disease can move in between patches on ocean currents. And so what we've done is nested many thousands of these compartment models, these SIR models, into this metapopulation context, this connectivity network. And in the Florida Keys, for example, we've got about 17,000 independent, well, not truly independent, 17,000 SIR models that are connected through the dispersal disease. We've got a couple papers that have come out from Florida. The first one was the first time that we had nested these models into a connectivity network. And we were able to predict the progression of the disease down the Florida Keys with fairly high accuracy, which allowed us to say with some certainty that we believe that indeed the pathogen is waterborne and dispersing on currents. We also did some tests and were able to show that whatever the disease is associated with, whatever kind of pathogen it is associated with neutral, well, it's dispersed neutrally buoyant, as a neutrally buoyant particle. So either it's neutrally buoyant itself in the water column, or it's associated with a neutrally buoyant particle. We also were able to show that the model predicts that corals should be getting this disease, infecting others, and dying very fast, which is what we see in the field. We also were able to model the dispersal of this disease out to the very tip of the Florida Reef Tract out at the Dry Tortugas. It took quite a long time, actually, for the disease to get there. And we were able to show that that's because it takes a very unique hydrodynamic environment to be able to move this disease to the west, out to the Dry Tortugas. It has to do with the shedding of eddies off of the Florida current. And I mention this because our model actually suggests that the disease probably got there a full 5 months or so before the disease was first recorded by the national park. And so, where's it a sanctuary? I think it's a park. And so our argument here is that we should be running these models sort of all the time, because we might have been able to not only notify management to go look for the disease and potentially mitigate it, because you can use antibiotic ointment to slow these lesions. but not just when to go, but where, because this is a very high resolution model. And so we may have been able to actually predict what portion of the dry tortugas were initially affected. So we were able to show the dispersal of this disease hydrodynamically down the Florida reef tract. If you think about it, that makes a lot of sense. This is a highly linear reef, right? But in the Virgin Islands, we've got a much more complex island archipelago here, and the initial dispersal of the disease was also much more complex. So we're currently working on getting a very similar model working in the Virgin Islands. We recently acquired a new hydrodynamic model, which we're excited to apply to this problem. This hydrodynamic model has hourly outputs, so it's got tides in it, and you can see the flow is very complex around these islands, and we're excited to see if our model works when we include this complex flow. We're also getting into the field and assisting also some of the other researchers on these programs with field collections. And we're trying to fit these SIR models to actually field collected data. And so what you see here is different locations in the Virgin Islands and observations of disease prevalence. And my student, Ben Farmer, is fitting SIR models to those observations so that we can try and get those rates, so the rate of infection, rate of mortality, for example. And he's finding that there is a lot of similarities. So we're finding that sort of good ranges for these parameters when we look in different places, but actually that each reef seems to have slightly different rates. And this kind of makes sense because remember, this disease affects over 20 species of coral, and that those corals have different susceptibility and infectivities and mortalities. And so we think that community is playing a big role in local disease trajectory, which we then think propagates out to the sort of metapopulation or meta-community context. And the theory here is that these high susceptibility species like this brain coral get the disease and they die very quickly, but in the process infect their neighbors that are lower susceptibility. Those neighbors take longer to die, but while they're infected, they're shedding this disease into the water column, which gives more and more opportunity for that disease to be transmitted to another patch. So I'm going to focus on the center panel. Excuse me. What we think is happening is that there's high susceptibility species disease initiation, and then low susceptibility disease maintenance happening. And we think this is why this outbreak has gone on for over 8 years, because these low susceptibility corals are sort of maintaining the duration of this disease. So what this means is that we have to develop a much more complicated SIR model. And this is what we were funded by NSF to do. And so through the EEID, we are looking at community on multiple scales. We're looking at coral community, microbial community, and endosymbiont community, and how that influences susceptibility and mortality of these different corals. We're also looking at predator community, because there's some evidence that coral predators like butterfly fish can peck at the infected tissue and then carry the pathogen in their bodies and then poop it out on healthy corals nearby. And so what we're doing is we're building a much more complicated compartment model where we have different categories of disease susceptibility, all of which can infect each other, but at different rates. And those corals can also are removed from the system at different rates. And of course, nested within this connectivity network. We have this model running in the Florida Keys. We have a paper that we hope to submit very soon using this more complicated community-based SIR model that does what we expect it to do. We just need to continue parameterizing it with field collected data. And we're just starting to do this in the Virgin Islands, where we are just getting started on modeling the initial progression of the disease and making sure that our model matches up with reality so that we know it's doing the things it's supposed to do. Next steps, as I mentioned, is incorporating community on various scales into these models, trying to understand how spatial heterogeneity in community is influencing the movement of this disease, and how it's influenced the duration of the outbreak and endemism. I also want to mention briefly that Ashley Rosin is doing a ton of histopathology. She's looking at how different species respond at the tissue level to this disease, looking at what makes different species more resistant to this disease than others, and also looking specifically at the process of disease-related dysbiosis, because actually at the tissue level, this disease actually looks quite a lot like bleaching, although accelerated very fast. And it looks like what's happening is that the coral and its endosymbiont are having a little bit of a battle, and essentially the tissues of the coral that surround the symbiont are experiencing extreme necrosis. So something is going on at the, right in that relationship of the endosymbiont and its host. So to sum up my talk, we're looking at, or we're trying to use metapopulation models to anticipate the influence of environmental change on persistence. We're doing that for corals, definitely, but we're also going to be doing it for stone crabs. But also retention is essential to local population persistence. So we're developing these high resolution biophysical models to understand how the physical environment and the biology of coral reef larvae interact to result in locally persistent or resilient populations. Both of these scales, there are implications for habitat protection or restoration, the management of fisheries, that's fish, shellfish, etc. And coral disease is a super bummer, but we are developing a suite of modeling tools that we really hope will be useful, hopefully in the mitigation of this disease, but maybe also in the mitigation of future marine diseases.